as we continue our focus on looking at small molecules and the roles they play in chemical biology, we are going to transition from our last few discussions of quorum sensing, a mechanism by which microorganisms use small molecules to communicate with one another and coordinate behaviors, into looking at chemical probes. Chemical probes are where we use small organic molecules to explore biological phenomena, understanding the mechanisms of those biological phenomena. In other words, how do those processes occur at the molecular level? And we can also use these small molecules called chemical probes to assemble biochemical pathways. The way that these chemical probes work is generally by binding to some protein that is involved in a biological phenomenon or involved in a biochemical pathway, and by binding to that, enabling us to detect that particular protein and either activate or inhibit it, we can recognize that particular protein's role in the broader biological process. So to write a little bit of this down on paper here for us, when we're looking at chemical probes, what we are evaluating is that we're looking at small molecules, generally small organic molecules. And much like with our metabolomics definition, generally small organic molecules are ones that have a molecular weight of 1,500 Daltons or less. And so we're generally talking here not about proteins or complex carbohydrates or things like that, but instead more like the traditional synthetic organic molecules or natural molecules that are similar in structure to drugs. So they're small organic molecules that are used to elucidate or determine the molecular basis of biological phenomena. So we're trying to understand at the molecular level, at the level of the genes, the proteins, and the other biomacromolecules, how does life function? What is the molecular basis for phenomena that we could observe, such as cell growth and cell division and things like that? How is that occurring at the molecular level? We can use small molecules to understand that. And also, another common use of these small organic molecules as chemical probes is not only to elucidate the molecular basis of biological phenomena, but also to, as well, assemble biochemical pathways. So, for example, to understand how proteins are assembled, if we can block certain steps of the pathway and freeze the process at that step, that will give us additional information about that particular step or the ones leading up to it to help us assemble that full biochemical pathway, the multi-step process by which complex molecules such as proteins or other biological molecules are assembled. So we're using here and relying upon these small organic molecules as being effectors of biological systems. And ideally, for a compound to be useful as a chemical probe, it needs to affect some specific aspect of a biological system. So when we say it affects a specific aspect of a biological system, it needs to affect a specific protein, for example. There are a variety of small molecules that, rather than impacting just one protein, for example, within a cell, instead they impact a variety of biochemical processes and a variety of different proteins within the cell. And that would make that particular compound not be particularly useful necessarily as a chemical probe, because if there are several different proteins that that particular small molecule is interacting with, it becomes a very complex problem to address what the impact of each of those effects is. So we're trying to look for small molecules that have some specific target within the cell, be it a protein target or something else that they interact with and therefore create some effect within the cell, such as inhibiting a biochemical pathway or altering the process of cell division, looking at biological phenomena that these small molecules can impact. Much like in the last video when we were talking about where do we discover new drugs from, Specifically, we were talking about where do we discover new quorum sensing inhibitor drug candidates from. The ways that we discover chemical probes are similar because there's a lot of parallels between the drug discovery process and the process of developing and implementing chemical probes. So we can answer the question of where do chemical probes come from, or how do we design and discover chemical probes. One of those ways is through high throughput synthesis, which, much like with the high throughput drug discovery that we talked about in the previous video, this is the strategy where a variety of different compounds, thousands of compounds, are synthesized and then evaluated for biological effects. In the case of using these types of compounds as chemical probes, one would pick some set of those particular molecules, and they would use those to evaluate what particular process within a cell they modulated, that they affected. And generally, one would have some hypotheses about particular classes of compounds that might be hypothesized to interact with certain classes of proteins and inhibit or activate those proteins to study the role of those particular proteins within the cell. So we'd be looking here with that high throughput synthesis for compounds that had impact on a particular type of protein within the cell so that we could understand the role of that protein within the cell by inhibiting or activating that protein using the small molecule. Another strategy for developing chemical probes is the targeted synthesis of compounds that are predicted to inhibit or activate a particular target, a particular protein within a cell. So targeted synthesis of compounds predicted to inhibit or activate a specific protein or a specific class of protein within a cell. And this we could refer to as structure-based or structure-guided chemical probe design. So this would be prevalent in cases where we had a specific protein in mind, and that protein had an unknown function within the cell, but we hypothesized that it was important for some biological process like cell division, for example. And so in order to investigate the role that that protein actually plays within cell division, we would design compounds based on the structure of that protein to inhibit the protein. And by inhibiting the protein, we would then monitor what was the ultimate impact in the cell. Was cell division frozen at a particular stage to understand what the role of that particular protein that we had inhibited with a small molecule was in the cell division process or in some other biological process. So if we have a particular protein in mind that we wish to study and explore the role of, we can use the structure of that protein to guide the design of compounds that are expected to bind to that protein and inhibit or activate that protein in order to then look at the broader biological effect of what does inhibition of that protein do to the processes that are occurring within the cell. And then the third place that chemical probes come from is from natural products, molecules that are isolated from living things, which, much like natural products, form greater than half of drug molecules that are on the market today. Natural products, organic molecules produced by living things, likewise have been really valuable as chemical probes. And one of the readings that you have available to you this week is a review article by Aaron Carlson, who reviewed natural products as chemical probes, providing a survey of a variety of different applications of these natural product molecules as chemical probes. And in a later video, we're going to get into a specific research paper that utilized a compound called amosamide to understand steps of the cell cycle, and amosamide was a natural product as well. So we'll use that paper in an upcoming video as a specific example of how chemical probes are developed and how they're applied. For now, we're sticking in this video with more general information about overviewing where do chemical probes come from, with the three main sources of those chemical probes, and what do we use chemical probes for. So with some understanding of where we can develop chemical probes from, what are the methods for designing these small molecules, the next thing that we need to look at is how do we track where those small molecules are going within a cell? Because it's one thing to come up with an organic molecule that enters the cell and has a biological effect, such as inhibiting cell growth or inhibiting cell division. It's another thing to go in and determine exactly why is that small molecule having the biological effect. And understanding the molecular basis of why a biological effect is occurring is critical not only toward understanding the basic fundamentals of how cellular processes work, but is also extremely important in the process of drug discovery, because in drug discovery, generally before a drug goes on the market, it needs to be very well established why that molecule is impacting a living system. In other words, why does a molecule act as a cancer chemotherapy? So generally in the drug development process, we're tightly intertwined here with the concept of chemical probes, because just like with chemical probes, we are trying to use small molecules to understand how a living system works in a fundamental sense with chemical probes, and then in the applied sense, using those compounds to inhibit particular biological targets that create effects that are beneficial toward life and beneficial toward organisms during the drug discovery process. So let's take a look at the methods that we can use to create chemical probes, to create tractable small molecules that we can trace where they are going within a cell, so that we can use those as tools for evaluating biological phenomena and things like that. So when we think about how are we going to detect and track chemical probes within a cell or organ, in other words, how are we going to use that small molecule to determine exactly where it binds within a cell? In other words, what protein does it interact with, for example? So in order to do that, generally the small molecule itself is not going to be suitable for enabling us to track its exact whereabouts in the cell. That becomes a very complex problem to take a standard typical small molecule and sort out at that molecular level exactly what protein in the huge plethora of proteins within a cell is that particular small molecule going to and binding with. And so in order to accomplish this question, answering this question of what particular target protein within a cell is a chemical probe interacting with, generally what we have to do is take our initial small molecule and modify that to make it detectable as a probe, so we can track exactly where that small molecule is going within a cell, and hence track what particular biomolecule in the cell, such as a protein, is that small molecule binding with and interacting with and inhibiting or activating. So generally what we're going to see is that small molecules that we're using as chemical probes typically are going to require some level of modification in order to track their whereabouts within a cell. So if we want to pinpoint exactly where a particular small molecule goes within a cell to identify its protein target, we generally need to make some sort of modification to that particular small molecule so that we can visualize where it is winding up within a particular cell. And so some common modifications that are executed with small molecules is that the small molecule is subject to a chemical reaction in order to attach some appendage on it that makes it traceable within a cell. And so what's generally done here is if we think of this square schematically as we call it the bioactive molecule because it's binding to some protein within a cell, and we are trying to discover exactly what it is binding to. Generally, that small bioactive molecule has to be covalently tethered to some portion of the molecule that has been appended to that is modified so that we can detect the small bioactive molecule. So we can think of this chemical modification as being a tracer group, a group that allows us to visualize where the small bioactive molecule is going, because due to the fact that these two are covalently tethered, wherever the small bioactive molecule is going is where the tracer will go as well. And so what are those common chemical modifications that are done to create molecules that can be tracked within a cell? Well, one common modification is that star that we've represented there as our tracer represents a fluorescence label. So we can add functional groups onto molecules that will result in that molecule as a whole exhibiting fluorescence. And by exhibiting fluorescence, we can then use microscopy techniques to image exactly what part of the cell is that particular compound going to. So we can narrow down to the level of something like the organelle to determine where a particular compound resides. So we could, for example, take a bioactive small molecule that we know inhibits cell division, and we could attach a fluorescent tag to that small molecule, supplement cells with that small molecule that's tagged, and then use microscopy to look at where that fluorescence ends up, and it might end up in, say, the mitochondria. And in that case, we would say, okay, the process, the particular stage of cell division that's being inhibited here is going on within the mitochondria because that's where the fluorescence is occurring, indicating that that's where the small bioactive molecule is gone since that had the fluorescent tag attached to it. So there are a variety of different fluorescent functional groups that can be covalently tethered to the small bioactive molecule. Another approach here that is taken is that a radio label can be used. And much like attaching a fluorescent label, where we're bringing in a covalent bond here to the small molecule with a chemical modifier, rather than this star representing a group that fluoresces, instead this star would represent a group that is radio labeled. For example, rather than containing carbon-12, it could be a chain of carbons that contains carbon-14, because carbon-14 is a common radioactive form of carbon that we could covalently tether there. And then using radio imaging, we could detect exactly where this carbon-14 label ends up within a cell, coupling that perhaps with microscopy to look at where what organelle the radio label appears within, and use that as our way of tracking where the small molecule has ended up within the cell and what that small molecule's target is. Then another common approach is to chemically modify the bioactive small molecule in green here with a chemical modifier or a tracer that is a group that contains an affinity tag. So we can attach an affinity tag with a common example of an affinity tag being the compound biotin. And the way that this use of an affinity tag works is that the compound is covalently tethered to biotin. The biotin allows the compound, the small bioactive molecule, to link to avidin, which is a common chromatography packing material. So what happens here when we use the affinity tag is in the use of the affinity tag, we would take our small molecule, form a covalent bond between that small molecule and biotin. So I'm going to put small molecule plus biotin, react to give a small molecule that is now covalently tethered to biotin. And by the way, there are a variety of commercially available kits that have created various derivatives of biotin that have different functional groups on those biotin molecules, so that those functional groups on the biotin molecules we can tailor to react with specific functional groups on our small molecule. For example, biotin, one functional group that can be found on biotin and its derivatives are reactive carboxylic acid groups that we could use to covalently tether to, say, an amine group in the small molecule to link those two together. So we create a small molecule that is covalently linked to biotin. And then what would happen is that small molecule that's covalently linked to biotin is provided to a cell. Specifically, this is often done with a cell lysate, meaning that we have broken open the cell, released the intracellular contents, meaning the mixture of proteins that are found within the cell and other biomolecules. So we provide that small molecule biotin conjugate to the cell lysate. And what's going to happen when it's provided to that cell lysate, if things go as we hope they will, is that the small molecule will enable the structure to bind to its protein target. So it binds to its protein target within a cell. So it's being tracked to that protein target, whatever protein it's impacting within the cell. And then at that point, what you have is a complex that contains the protein target that is complexed or strongly interacting with the small molecule that is tethered to biotin covalently. And then this particular complex will ideally, when applied to a column that is packed with avidin, which biotin has a really strong affinity for, this particular complex will bind to the avidin column, whereas all of the other biomolecules, the untagged proteins, will continue through the column and not be strongly bound to it. So at this point, after we enable that complex of the protein target to form with our small molecule that is linked to biotin, then that mixture of proteins, including the one that's bound to the small molecule as well as all the other proteins from the cell lysate, are applied to a column. That column is packed with avidin. Avidin very, very strongly interacts with biotin. And so what's going to happen here is that the protein that is interacting with that small molecule chemical probe that is covalently linked to biotin will bind strongly to this, remaining tethered, remaining attached, adsorbed to the avidin, whereas all of the other proteins within that cell will continue through the column. And so at the end of that process then, what one can do is release the avidin biotin complex and then identify the protein through mass spectrometry or whatever your favorite technique is for identifying a protein, determine what the particular protein was that was bound to the avidin column and interacting with the small molecule that was tethered to the biotin. So then we can ID that protein that was interacting with the avidin. And it's interacting with the avidin because of the fact that that biotin label is there. And with that, then that would reveal the protein target of that original small molecule chemical probe. And so in this way, we can sift through and sort out exactly what portion of a biological process that small molecule is impacting, because we take the small molecule, we link it to a tracer molecule, in this case biotin, our affinity tag that makes the covalently complexed molecule strongly adsorb to avidin, and that way we can separate out the protein that is interacting with the small molecule and biotin from all of the other milieu of things in the cell to ultimately identify that protein that is complexed with the small molecule and biotin, so that we can determine the structure of that protein and link it to the observed biological effects such as inhibition of the cell cycle or whatever else might be the case. So in these common modifications, everything sounds well and good on paper, but things always get a little bit sideways when actually going into a lab to carry these out. And so I want to mention some of the common challenges that are associated with this, because I like to think that science occurs in a nice, clean environment. Everything occurs like we hope it will. But there are definitely some challenges to this. And so challenges that are associated with chemical modifications include, first off, the requirement here is that we be able to covalently link a small bioactive molecule to the tracer, to the fluorescent label, the radio label, or the affinity tag. And depending upon what functional groups are present in that small bioactive molecule, that might be a challenging thing to undertake, because if there aren't reactive functional groups within this original small molecule, it would be very difficult to create a covalent bond to the chemical tracer. And so challenges include that the chemical covalent tethering to those tracer groups requires that the original molecule have reactive functional groups. So it requires that the small molecule have reactive functional groups. And so if we don't have the appropriate functional group or groups present within the bioactive small molecule, we aren't going to be able to link it to the chemical tracer. The other related problem at the other end of the spectrum is that if you have a bioactive small molecule that has a whole bunch of different reactive functional groups that will all react with the chemical tracer, then you could end up with a bioactive molecule that has a bunch of tags on it in a bunch of different places. And if a small molecule is too heavily modified, its structure has been dramatically altered, it's no longer going to be able to interact with its protein target. And so we need, ideally, a tracer group that we can covalently tether at one specific targeted location to the bioactive molecule. And that's not always the case, depending upon what functional groups are in that small molecule. So ideally, we need one functional group in the organic molecule that reacts with the tracer, so that we can attach that tracer, the fluorescent label, the radio label, or the affinity tag, to a specific targeted location of the organic molecule and create a product of that reaction in high yield, where we've yielded one specific product. Because otherwise, if you have labels on a variety of different portions of the molecule, that might result in the compound no longer binding to the target. And that brings me to the next challenge, is that the small bioactive molecule has specific regions of that small bioactive molecule that enable it to interact with the protein target. And so when you attach covalently this chemical modifier onto that small bioactive molecule, if you are interfering with the portion of that bioactive molecule that's responsible for the bioactive molecule interacting with a protein target, then you are going to obliterate the ability of that small bioactive molecule to bind to the protein target. And so another challenge of these modifications is that the covalent linkage of the tracer may inhibit or halt the small molecule from binding to its target. So in other words, how this could be observed would be that a scientist would observe that the small bioactive molecule had an intended biological effect when originally tested. Like, they incubated the small bioactive molecule with some pathogenic bacterial cells. They observed that the bacteria cells died, suggesting that this compound was an antibiotic molecule. They wanted to understand then, how does that small molecule act as an antibiotic? And so they decided to tag that small bioactive molecule with a fluorescent label or a radio label or an affinity tag. Then after they incorporated that tag, they provided that small bioactive molecule that's tagged to the bacterial cells again. And suddenly now the bacterial cells are no longer inhibited by the compound, because the compound now has that tracer on it. And that would indicate that the linkage of that tracer to the bioactive molecule is preventing the bioactive molecule from binding to its target any longer. And so that would then be a failed experiment, because with the tracer in place, the compound is no longer able to interact with its target and create the desired biological effect. And so these are pretty common challenges that have to be overcome when dealing with creating chemical probes and testing those chemical probes, is that the chemical tracer that we wish to attach may not be compatible with the functional groups that are in the small molecule, meaning that we can't get a reaction to occur between the tracer and the small molecule, or that the tracer covalently links at several portions of the molecule, which is not necessarily desirable either, or that when we link the tracer to the small molecule, then the biological effect no longer occurs. It's no longer able to bind to its target because the structure of it has changed too dramatically. So those are things that would send us back to the drawing board, back to looking at alternative approaches to developing chemical probes. Unfortunately for us, with the fact that there are multiple types of common modifications, the fluorescence, the radiolabeling, and the affinity tag, those are compatible with a variety of different functional groups to facilitate strategies for covalently linking those tracers to the small molecules that we intend for them to. And then as a result of the fact that there's a variety of different portions of most organic molecules that we can tag, if at first we don't succeed, and if at first when we covalently link a tag to our molecule, it no longer binds to its target, we can look at a different region of that small molecule and try attaching the tag there, and then retesting to determine whether attaching the tag at a different location, a different functional group within the molecule, then allows it to bind to its target. Because in many cases, when we look at small molecule drugs, there's a particular region of that drug molecule or that chemical probe molecule that confers the biological activity. If we leave that portion intact, then the molecule will be effective even if we attach a tag or a tracer to some other portion of the molecule. So what we are going to do now in the next video is we are going to take what we've learned here and we are going to dig into a specific example paper from the literature where the biological target of a compound called amosamide was discovered to identify unique aspects of the cell cycle in the development of cancer chemotherapies of the future.